Hello, my name is Stephanie, and today I will be explaining what genetic modification is and what steps are involved within the process.
Genetic modification is an efficient process which is fast and cheap and can be used for industrial and medical processes.
So initially, let's brush up on some background information that will be required for this topic. Let's start by defining what a gene is. A gene is a heritable factor that controls a specific characteristic, such as hair color, eye color, skin color, etc. The genetic code is universal, which means that genes can be transferred from one organism to another, even if the organism the gene was taken from is from a different species than the one the gene is being placed in. Organisms that have genes transferred to them are called genetically modified organisms. The process of transferring genes is called genetic modification. So now let's go through the steps of genetic modification. In this case, I will be using the example of the technique used to produce insulin, where E. coli bacteria is used as a host cell. So initially, one isolates a cell from the area of interest, so we'll be isolating it from the pancreas. Then a gene of interest is isolated, in this case insulin. Then DNA bases are added to the ends of the insulin gene to make sticky ends. In this case, we'll be adding guanine and thymine. Then a bacteria cell is taken, and its plasmid is removed. Plasmids are small loops of DNA that are found in bacteria. Then the plasmid is cut using restriction enzymes. Then, on the part of the plasmid that will be used further on in the process, sticky ends are added. But now, instead of adding guanine and thymine DNA bases like we added on the insulin gene, we'll be adding their complementary bases, which are cytosine and adenine. So when the insulin gene and the plasmid are mixed together, they link by complementary base pairing, what happens between guanine and cytosine and thymine and adenine. Then, DNA ligase is used to seal the gene, the insulin gene, and the plasmid together. And the plasmid and the insulin are now called recombinant plasmid. A suitable host cell is then found, in this case E. coli bacteria was used, and the recombinant plasmid is placed within it. E. coli is cultured and starts producing the human insulin, which is extracted and purified and used for diabetics.